Okay, welcome Science 10 students to the review of transcription and translation, a summary of what you did last class when you were completing the assignment on transcription and translation terms. So this is an overview of both the transcription and the translation piece, and we can separate the two into two sections here, where the first section is transcription, and the second section, translation, where the whole, as a total, transcription and translation form a process called gene expression. So taking information from DNA and turning it into proteins. That is the general idea here. Now, in transcription, we have this, that within the nucleus, a RNA polymerase will bind to the DNA strand. So then RNA polymerase is going to bind in here onto the DNA. And with the help of something called the helicase, it will rip the two strands of the DNA apart, where it then creates an RNA. So zooming in here. So this is the RNA strand gets created from that DNA. And it gets processed a little bit more in order to create mRNA, which then leaves the nucleus to go and translate. So this whole first part where we're taking information from the DNA, turning it into mRNA, is called transcription. Okay, so transcription, reading the DNA, creating mRNA. All right, next piece. We have this mRNA. So this is the beginning of translation now. mRNA has left the nucleus. It comes in contact with a ribosome. A ribosome is split up into two different units that latch onto the mRNA. And then the ribosome acts essentially as like a workbench for the tRNA to come in and bring amino acids in order to create a polypeptide chain. So the ribosome just kind of acts as the site where translation can happen. Where mRNA comes in here, it gets read by these tRNA. So if the mRNA is moving this way through the ribosome, the tRNA, so these molecules with an amino acid resting on the top, comes in here, attaches to the mRNA strand, and it attaches to specific codons. Remember that codons are a sequence of 3 bases, and the tRNA has the anticodon in order to attach onto that codon. And then once that happens, it leaves this amino acid behind, okay? And then leaves, allowing for the next transfer RNA to come in here and attach the new amino acid to the old amino acid. So in this case, to the first amino acid. So as you can see, the tRNA continue to come in here and attach to the next codon, and what gets created is this polypeptide chain. And in here it says a protein chain. Well, a polypeptide is a string of amino acids. Some polypeptides will just act as proteins for the cell that the cell uses to function in different ways, and some polypeptides will form with other polypeptides in order to create more complex proteins, which would do a different function. And so this, in all, is transcription and translation, and the whole point of it is to create proteins for our cells. And the next thing to do is discuss how this, or kind of turn a codon, so what specific codons program or code to what specific amino acids, and that's the next step. So that's the summary, and move on to the Google Classroom assignment next.